Hello everybody. In today's video, we're going to be talking about the cell cycle and mitosis through a couple of processes. I want to start off with some examples, so let's check them out real quick.
All organisms have some level of regenerative abilities. For some animals, regeneration is just a part.
We'll talk about that guy later. And I do want to mention, guys, that all of the links to all the videos and the slideshow will be available in the description.
Of life. Flatworms and hydras can regenerate their whole bodies, even from a tiny piece of their original selves. Starfish, octopuses, or even crabs can lose an arm and grow it back without much of a problem. But these animals all share something in common. They're all invertebrates. Invertebrates like us, regeneration is pretty much limited to regrowing skin or scar tissue over a wound. Except for our liver, we can't regrow our organs, nor our limbs. In fact, almost no vertebrate can regenerate much beyond what we can. That is, except for the axolotl and the other members of the salamander order. The only other vertebrates that can regrow limbs are frogs, but frogs lose the ability once they fully turn into their adult form. The axolotl retains its regeneration ability throughout its entire life. The way an axolotl regrows a limb begins with the limb's surviving cells. Once the limb has been severed or lost, a blood clot rapidly stops the bleeding at the cut, and a layer of cells covers the area of amputation. This part is not so different from the way we would heal. Then, over the next few days, the cells underneath the epidermis begin to rapidly divide, forming a cone-shaped structure known as a blastema, also called the regeneration bud. The blastema is what holds the key to regeneration. It's where regular bone, cartilage, or muscle cells de-differentiate, meaning they lose their identity and transform back into stem cells. Slowly, they start reforming bone, skin, and veins, much like stem cells would do when the animal is first developing in its egg. As the cells in the blastema continue to divide, nerves and blood vessels connect it to the rest of the body. The limb eventually grows back completely, looking like an exact copy of the limb that was lost. And while total limb regeneration is impressive enough, axolotls are also capable of something that seems like it should be downright impossible: regrowing parts of their brain.
Alright, guys. So that's the first one. Here's the second one I want to show you.
Okay. Seriously. I promise. It doesn't even hurt. That would be curtains for most organisms. I mean, this piece here is just a chunk with no head and no tail. But watch. You can see under a microscope that overnight its wound heals closed. Okay, maybe we could do that.
Noticing any common trends here?
More slowly, of course. But then it starts to regenerate, growing new tissue. It's that white part, called the blastema.
So that's also what the other one did.
In a week, see those tiny little spots? They're new eyes, grown from scratch. But the planarian, can I call it that yet? It still kind of looks like a blob. By day 12, it's starting to look like a proper planarian. It's got a head, an
Notice that the new tissue is doesn't have pigment yet.
entirely new one. And a new tail. And it's doing its regular planarian things, like pooping. That's because one of the first things it started regrowing was a tube called the pharynx, which is how things go in and out. It sucks up food, like this beef liver that scientists are feeding it. After 3 weeks, it looks
Alright, so, with that out of the way, what do all these things have in common? Getting a cut that we showed, we showed the after process in that one video, the first video. The reproducing by cloning via fragmentation, so that was what we saw with the second video with the planarian. Regeneration, so we saw that with the first video with the axolotl. And embryonic development. And I do include this video in the slides. I just think the other two are more impactful. So if you want to go back and watch that, you can. But kind of think about the process of embryonic development and what that would entail. And kind of think about what all these things have in common. And if you were paying attention at the beginning, you might have an idea that that thing is going to be mitosis. So we're going to talk about mitosis. So here's some interesting facts I think you guys should know. And there will be a link to a worksheet that you guys can fill out along with me if you choose to download it and use it. Also, if you want to use this as like some kind of supplement for classroom or homeschool, you could also use it here. So, some interesting facts. Over 95% of all cell division in your body is mitotic, meaning it's using mitosis. So the other 5%, of course, is going to be meiotic, and that's going to be meiosis, and that's going to occur only in the reproductive organs. So around 330 billion, with a B, new cells are made or replaced in your body every day. So that's kind of astounding. It's pretty amazing. That's going to be about 3.8 million a second. So most of these cells are going to be in the blood, in the digestive lining, so like the esophagus and the small intestine, large intestine, skin, and some muscle, much more slowly of course, along with bone being slower than muscle, and then the slowest is going to be nerve tissue. So those types of cells are replaced much slower. Seen in the videos I showed you, mitosis is the underlying process behind regeneration, cloning in some animals, growth and development from an embryo, wound healing, like what we do, and maintenance. So maintenance is super important. It doesn't get thought about, doesn't get mentioned enough, but maintenance is going to be how we keep going. If we didn't maintain our body or maintain our cells and replace them, we wouldn't live past a month or more, maybe 100 days, because you replace about 1% of yourself every day. So that's super important, and that's why what we eat is super important. So whenever you think about it, if you feel like crap all the time, and all you're eating is pizza and drinking soda pop, think about, we have to fuel these processes so that we can maintain ourselves and grow. And if we're not fueling the processes properly, we're going to have a hard time having energy to fuel them and then have energy for us. So that's why it's super duper important with what you eat. In bacteria and microorganisms, this process is the method for asexual reproduction. So the A in asexual means without, so without sex, and they reproduce in a process what's known as binary fission. So essentially, the bacteria or the microorganism is going to simply divide in half, and it's going to make a copy of itself. It's going to be an identical copy. The cell cycle is going to be broken up into 6 key steps, which we're going to outline in detail in the next slide. Those steps are going to be interphase, prophase, metaphase, anaphase, cytophase, and no, telophase and cytokinesis. There's not a cytophase. Telophase and cytokinesis. So by the end of this lesson, I'm hoping that you'll be able to identify uh, these diagrams or these models here, and be able to name them and be able to explain what's going on. And by explaining what's going on or being able to identify what's going on, um, you'll be able to recognize them and know them. So interphase is what's known as the preparation phase of the cell cycle. It's not really a part of mitosis, but it is part of the cell cycle. It itself is broken up into 3 different subparts. So if you think of the cell cycle like a clock, you can see there's 6, 12, 18, 24 hours. Most of the cell cycle is going to be spent in interphase, with only a little bit, about 10% really, is going to be spent in the mitotic phase, which is what the rest of this lesson is going to be about. So the first part of interphase is going to be what's called G1. It's also known as gap 1, but what I want you to remember is that G1 stands for growth. And during this part of interphase, the cell is going to grow larger in size. It's also going to perform normal cell functions. So whenever you see or think about, you see a diagram of a cell, or you see a cell model, what you're normally going to be seeing, and you don't realize it, is you're seeing a cell in interphase. That's because all the normal functions, like making proteins, changing glucose into energy, and all the other like waste, transporting waste and food, is all going to be done during G1. So that's going to be a big chunk of the cell's life, about 25%. The cell can also leave the cell cycle or repeat G1, meaning it's going to decide not to divide. So it can spend a longer duration in that process. This is known as quiescence, and that's also what a lot of our nerve tissue and bone tissue are going to be doing. They're not going to be dividing constantly like our skin. They're going to be staying in that regular life phase where it's just doing cell stuff. It's not really replicating DNA or dividing. This kind of brings me into the second part, which is called S phase. The S is going to stand for synthesis. This is when all the DNA replication is going to happen, and essentially what DNA replication is going to be is just making a copy of the DNA. So let's say you've got a test coming up, and it's 3 days away, or 2 days away rather. What are you going to be doing the night before the test, or the second night before the test? Well, hopefully the answer is going to be you're going to put all your energy into studying for that test. Well, the cell is really going to do the same thing. The cell is going to put all of its energy into making a copy. So let's say you missed 2 weeks of class, and you've got a buddy, and they took perfect notes. They took great notes over the class. You're going to spend all your energy making a copy of those notes. So the cell is going to make a copy of its DNA. And all cell functions that are normally talked about are going to be put on pause during this phase. The last part of interphase is what's called G2. It stands for gap 2. And it's going to be a second growth phase. So during this part of interphase, the cell organelles themselves are going to be replicated. So, for instance, your mitochondria, your Golgi apparatus, and the smoothie R are going to be made copies of themselves. The cytoplasm is also going to increase in volume. This is going to make the cell bigger again. And again, the mitochondria are replicated to provide enough energy for the cell to divide. Minor spoiler alert. That's what we're talking about here. Prophase is going to be the first real phase of mitosis. The pro in prophase stands for first, so think of prototype, or think of the prokaryotic cell. The prokaryotes came before the eukaryotes, so they came first. They're first. Pro, first. During prophase, the DNA that was replicated during S phase is going to condense from chromatin with the help of histone proteins into what we know as chromosomes. So from chromatin to chromosomes. And it bears mentioning that how we think about DNA, the double-stranded double helix, is it's a misnomer. It's what we think about when we think about DNA isn't entirely accurate. So I want to bring your attention to the top right corner here of the screen, where we see DNA, how we normally think about it. Well, it's not really like this. It's normally in a form known as chromatin, and what that's going to be is it has a double wrap around a protein known as a histone, and it's going to be bundled into a fiber, a densely packed fiber known as chromatin. So it's only going to be without those proteins, during S phase. And that's because it has to be in order to be replicated. So I will show you, hopefully if we have cooperation, I will show you this video.
In this animation, we'll see the
And this is going to explain how what it's going to look like when this condensation happens. The condensation from chromatin, or from bare DNA to chromatin to the chromosome.
remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result.
Guys, the video is really old, but it's really good. That's why I'm using it. But it is a little bit fuzzy. So.
Result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always.
So it's going to show you a little sneak peek of what's going to happen in the next couple slides. So pay attention closely.
Present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Okay. back to this. Oops. Okay, so back to what we were saying. The nucleus and the nucleus and the nucleolus. I still get tripped up. I still get tripped up with those, but the nucleus and the nucleolus are both going to dissolve and are going to be absorbed into the cell. So essentially here, the reason why this is happening is because they're not needed anymore. They're not necessary, because they're going to get in the way. So essentially, your nucleus, the nuclear envelope, is going to disappear. It's not disappear, but it's going to dissolve, and it's going to get absorbed into the cell because it's going to be needed later. Spindle fibers are going to emerge from the centrioles at the poles. So think of centrioles at the poles. That's how I remember them. And they're going to form what is known as the mitotic spindle. So that's the structure right here, the mitotic spindle. If you can see my cursor, it's right here on this diagram. And that's going to conclude what is known as prophase. So in metaphase, in the meta in metaphase means change in position. So you've probably heard the word meta before. It's not used the same way, but that's where that root word comes from, meaning change in position. So during metaphase, chromosomes are going to change their position and align on the equator or middle of the cell. And it really means down the center, not clumped in the middle, but down the center in a straight line. Centrioles move to the poles. So poles basically meaning extreme ends to left and right of the cell. Also want you to notice here that we also have change in position. See that phrase keeps coming up, of our organelles. So the organelles are going to change their position and be found on the left or right side of the cell. It's not going to be found in the middle anymore. There's going to be a opening, a gap forming where there's nothing but the chromosomes down the middle. The spindle fibers from the centrioles are going to extend until they attach at the chromosome at their centromere, and they're going to physically attach at a location that is known as the kinetic core. So, the top right, you can see here, there's a kinetic core structure on each replicated chromosome, and that's going to be the site of attachment for those spindle fibers. As we said earlier, the meta and metaphase means change in position. Those duplicated organelles are going to be positioned equally on either side of the equator, as I mentioned earlier. And remember, you can always pause the video if you need to write these, fill these blanks in, or just to take notes if you need to. So anaphase is going to be the next phase in mitosis. Of course, the ana in anaphase means apart. So ana for apart, ana for apart. So as you can see by my diagram here, that we've got this gap now in the middle of the cell where there's nothing but cytoplasm. Also, you can see here in the other diagrams, it's the same thing. Nothing but cytoplasm in the middle here. And what's going to happen is those spindle fibers that are attached to the kinetic cores of the chromosomes are going to retract. And as they retract, they're going to pull the sister chromatids to the opposite ends of the cell near the centrioles. So remember, the centrioles that are at the poles, well, they're going to retract those fibers, and it's going to bring everything with it. Also, I do want to mention at the same time other spindle fibers that are not attached to those chromosomes are going to actually extend, and they're going to push on adjacent spindle fibers with the help of what is known as motor proteins. So think of little motors essentially at the cell level, and they're going to start pushing on one another, and what that's going to do is it's going to stretch the cell further and further apart. And all this stress and tension is going to develop what's known as the cleavage furrow, which I'm highlighting here with my pointer, in the middle of the cell. And that's going to be where the stress is being put on the cell membrane. So, as my slide says, the compound action stretches the cell and develops what's known as the cleavage furrow, a depression in the cell membrane at the equator, or middle of the cell. Remember, you can always pause it if you need to. So, telophase or telophase is the final stage of mitosis. The telo in telophase means distant. So again, think of telephone. Tele meaning distant, phone or phono meaning sound. Or telegram, tele meaning distant, gram meaning drawing or writing. There's also like telemarketer, meaning someone who markets from far away, or does marketing. So there's many telos. During this phase, the nuclear envelope is going to reform. I mentioned earlier that it dissolved because it wasn't needed. Well, now we're going to need it again. So what's going to happen is it's going to repolymerize and form itself again around those two chromatids that are on either side of the cell now at the poles. The nucleolus is also going to reform inside the two new nuclei. The spindle apparatus on both halves of the cell is going to depolymerize or fall apart. So essentially, your centrioles are only really needed during the big action that happens during mitosis, and once that's over, they're really not needed anymore. So they kind of just fall apart and get absorbed back into the cell. So cytokinesis and telophase are often closely associated with one another because they happen so closely in time to each other. So some people list them as separate, and some people have them combined. I'm doing telophase and cytokinesis. Different people do it different ways. But cytokinesis is its own thing, but it happens right immediately after telophase happens. So what happens during cytokinesis is the cell is pinched at the middle and divides. And how this happens is there's what's called a contractile ring. So this ring right here. And what's going to happen, pardon me, is that it's going to get drawn shut like a drawstring on a bag. And it's going to pinch at the middle, and it's going to cleave the cell in two. In the in cytokinesis, the cyto is for cell. So if you think of cytoplasm, the cellular jelly, that's the cyto for cell. And kinesis, meaning motion, like kinetic. The kinetic energy is motion energy. Kinesis, motion. Okay. So I'm going to go through these real quick. You can pause the video at each one. I'll have a little pause, or I will pause myself, and then I'll give you the answer. This is also going to be on the worksheet, which I will attach in the description. Okay, so we're going to kind of go out of order here, just to give it some variety. So I want you to give a guess, either write it down or say it out loud, what we think is happening, and pardon me, is happening on H. I also want to mention, I want you to be able to tell me why. It's very important that we're not just identifying. We need to know why and explain it and be able to kind of defend your answer with evidence. It's part of the scientific process. So for H, go ahead and pause now. But the answer is going to be telophase or telophase. The reason why is because, as you can see, the two groups of chromatids are very far apart. They're on either extreme end of the cell. So if you remember, the telo in telophase or telophase means distant. So they're distant from each other. That's the easiest way to know. Okay. Next one, we're going to do G. So go ahead and pause. Give your guess what you think it is. The answer is going to be anaphase. And the reason why is because, as you can see here, remember, ana means apart. So anaphase, they're moving apart. So you can see the sister chromatids are moving apart from each other. They're separating. They're no longer lined up down the middle completely. They've broken apart into their separate halves. Okay. Next, we're going to do this one. I'll give a hint. It's not going to be interphase. It will not be interphase. But with that eliminated, what do we think this is? Go ahead and pause the video. The answer is going to be prophase. The reason why is that you can see the DNA has been condensed into chromosomes and they're not aligned at the middle, meaning they can't be a different phase. They're all scattered about. Also, you can see the mitotic spindle is right here. And that mitotic spindle gets developed in prophase. Okay. Now we're going to do I. This one's extremely well. I love that diagram. It's very well illustrated. It's very, you've got a lot of key things happening, and it's very apparent what's going on. So go ahead and pause the video and give me your guess to think as what you think this one is. This one's going to be cytokinesis. The reason why is because we've got the DNA has returned back into being, well, not the DNA, pardon me. The DNA is still in chromosomes, but you've got a clearly defined nuclear membrane or nuclear envelope happening here. So the white section is distinct from the green. So we can tell that we've already gone through telophase. And the other huge giveaway here is the cleavage furrow. It's super apparent. You can see the contractile ring here in the middle where it's getting pinched. So if you want to think of it as this is like the workout stage, it's like the dumbbell. If you see the dumbbell, you know, depending on who's asking the question, um, it's going to be telophase or cytokinesis. But here, it's cytokinesis because we've already talked about telophase. And we've already reformed, the nucleuses have already reformed. So it's after telophase. So it's got to be cytokinesis. Alright, that leaves only one left. We're going to talk about F now. So with all the other ones being mentioned, what else is there? What's left? So go ahead and pause the video, take a moment to think about it, and tell us what you think. The answer to this one is going to be metaphase. Remember, the meta means change in position. So we've got all of our chromosomes lined up more or less down the middle here. You could argue it's extremely early anaphase, but it really isn't. You've got your fibers here attached at the middles. Remember that centromere with the kinetochore. Is that going to be that? It's going to be that site of attachment for the spindle fibers. All right, guys. I will wrap up with a video, kind of going over the process again, showing you it happening, kind of live in motion, and then we will conclude the video, guys.
Cells go through different phases that include growth, DNA replication, cell division, and the production of two genetically identical daughter cells. This sequence is known as the cell cycle, which has two main phases, interphase and the mitotic phase, also known as mitosis. Interphase is the longest phase of the cell cycle, accounting for about 90% of the total cycle. During interphase, a cell recovers from previous division and carries out its normal functions. It grows in size by forming more organelles and replicating its DNA, resulting in the duplication of the cell's chromosomal matter. Next, the cell will go through mitosis, which includes division of the nucleus, and then the division of the cytoplasm, called cytokinesis. Mitosis also has several stages: prophase, metaphase, anaphase, and telophase, which are finally followed by cytokinesis. During prophase, the chromatin condenses into visible chromosomes, and the nuclear membrane disintegrates. The centrosomes move to opposite poles of the cell as spindle fibers start to form. The spindle fibers attach to the centromeres of the chromosomes. Metaphase is characterized by the alignment of chromosomes at the metaphase plate, an imaginary plane in the middle of the cell. The spindle
Remember the equator.
fibers ensure each chromosome is properly aligned. Anaphase begins when the sister chromatids of each chromosome separate and are pulled toward opposite poles of the cells by the spindle fibers. Once the chromosomes reach the poles, the cell elongates. During telophase, the chromosomes
Remember, in anaphase, those spindle fibers are going to extend as well as contract. So you've got some that extend and some that are going to contract, and that's what's really going to elongate that cell.
decondense, and new nuclear membranes form around each set. The spindle fibers disassemble and the cell prepares for cytokinesis. Cytokinesis, which sometimes begins during telophase, is the final stage of the cell cycle. Cytoplasm divides, which forms two genetically identical daughter cells. In animal cells, a ring of filaments forms around the equator of the cell. The ring contracts, pinching the cell membrane inward and dividing the cell. In plant cell cytokinesis, a cell plate forms.
All right, guys. So, if this were in a classroom, what we would do next is we would have a classroom activity where we could do a number of things, so I'm going to kind of mention them briefly. What we could do is you could use whiteboards as a group activity, and pipe cleaners, and essentially just divide your whiteboard into 6 equal sections, 6 equal squares, and as a table, you guys can basically kind of recreate the 6 phases of the cell, not mitosis, of the cell cycle. Then another thing that you could do is you could select one of the mitotic phases, pardon me, and recreate it using a paper plate as the cell, and basically make all the parts and kind of make a model of that phase. Another activity would be if you go in the hallway, you could have students reenact the cell phases, where they come together during metaphase, and then they run apart during anaphase. And there's lots of other activities you can do, but those are just a couple. I hope you guys enjoyed the video. If you liked it or learned something, let me know please in the comments. And hope you guys have a great day. And I will see you guys in a video coming soon. Bye guys.